System in general, the neuroglia, the nerve glue. Now let's talk about the actual neurons themselves, the structural units of the nervous system. These are going to be large, very specialized cells. They conduct impulses, electrical impulses within themselves, and they release neurotransmitters to talk to other neurons and other target cells. Their special characteristics include being extremely long-lived. They will last in a person's entire lifetime. The neurons we have when we die are the same neurons we had when we were born. They are amitotic, which means they will not divide. We cannot replace them. We don't have neural stem cells. There are a few exceptions on how they can be repaired. We'll talk about those a little bit. They require a continuous supply of oxygen and glucose. They have a very high metabolic rate. They are constantly using ATP up. And the brain and central nervous system, in fact, do not need insulin to keep glucose going into those cells there, which is a good thing if you're diabetic. They all have a cell body, and they all have little processes, at least one, coming off that cell body. So the cell body is also called the perikaryon or the soma, more often the soma or cell body. It's a biosynthetic center of the neuron. This is where proteins are synthesized, membranes are made, chemicals are formed. There is a lot of rough endoplasmic reticulum. That rough endoplasmic reticulum has been given special names in neurons. It's often called a chromatophilic substance, or it's often named after one of the scientists who first learned to stain them and make them visible, and that would be Nissl. So they're often called Nissl bodies, traditionally called Nissl bodies. They generally contain a spherical nucleus with a nucleolus. They sometimes have pigments, and the plasma membrane is part of a receptive region that receives input from other neurons. Most neural cell bodies are located in the central nervous system, and when we have a cluster of them in the central nervous system, like in the brain or spinal cord, we will refer to that as a nuclei. A similar structure in the peripheral nervous system will be referred to as a ganglia. The processes that come off, the central nervous system has both the neuron and cell bodies and their processes, while the peripheral nervous system is mostly processes that come off the central nervous system cells, with the exception of the ganglia that we will talk. So if we have a bundle of neuron processes in the central nervous system, we're going to call that a tract. If we have that similar bundle in the peripheral nervous system, that's what we're going to call a nerve. So that simple organ, the cranial nerves, the spinal nerves, are really like the tracts in the central nervous system. They're bundles of neuronal processes. Processes that come off a neuron are either bringing information to the cell body or taking it away. If they are bringing information to the cell body, they're referred to as dendrocytes. If they're taking information away, they're referred to as axons. So here's a typical peripheral motor neuron. So we've got a cell body here. These processes are dendrites. They are communicating with other nerves or other structures. This single one that goes a long ways away to a muscle cell here is called an axon. It is coated with numerous Schwann cells, giving you the myelin sheath. And these gaps, again, are called the nodes of Ranvier. They're going to speed up neural transmission. And the point where that axon comes off the cell body is referred to as the axon hillock. That's where we want to get an action potential to begin if this nerve is going to fire to its target organ or cells. So dendrites, most motor neurons, contain 100 or so of these short, tapering, diffuse branch processes. They have the same organelles you find in the cell body. These are receptive or input regions of the neuron. They're going to get information from messages send the information towards the cell body as graded potentials, which are short signals. There, in many brain areas, there are finer dendrites that are highly specialized, can collect information. They can contain dendritic spines or appendages with bulbous or spiky ends, but they're here. They're talking to other neurons and sending that information to the cell body. The axon, on the other hand, is how the neuron talks to other targets. It starts at a cone-shaped area called the axon hillock. Some neurons, axons, are short. In others, they're almost the entire length of the cell. They can be up to a meter long. The muscles in your toes are controlled by axons from cells that arise in your spinal cord. The longer ones we'll call nerve fibers. They occasionally branch, and those branches would be called axon collaterals. They branch profusely at their ends, their terminus. In fact, they can have up to 10,000 terminal branches, so they could be talking to 10,000 different target cells. We talked a little bit when we talked about muscles that one neuron could activate several different muscle cells. And their distal ends are called axon terminals or terminal buttons. The axon is the conductive region of the neuron. It generates a nerve impulse and transmits them along the axonema, which would be the membrane surrounding an axon, and it makes its way to the axon terminal. That terminal is the region where it secretes neurotransmitters. We saw that when we looked at muscles. In muscles, they released acetylcholine. Those transmitters are released into the extracellular space. They are not directly in contact with the cell they're talking to. Acetylcholine for us so far has only been an activating neurotransmitter. Neurotransmitters can actually excite or inhibit, especially when you talk to other neurons. With all these dendrites, we can be carrying on conversations with many different neurons at the same time. And with all these branches, we can do the same thing from a single neuron. So axons relay on cell bodies to renew proteins and cell membranes, but they will quickly decay if they are cut or damaged. Axons have an efficient internal transport mechanism. There are molecules, molecular motors, so that organelles can be moved along the axons and following cytoskeletal pathways, and they can move in both directions. So if something tends to move anterograde, it's away from the cell body, so we're going to take mitochondria and send them down these axons. Retrograde would be towards the cell body. Organelles that need to be degraded would be moved back towards the cell body and recycled. There are also some viruses, such as the rabies virus, that can take advantage of retrograde transport, and some other bacterial toxins that can do the same thing. So again, certain viruses and bacteria damage neural tissue by using retrograde axonal transport. The polio virus does this, rabies virus does this, herpes simplex virus, and the tetanus toxin from bacteria. And there is research underway to investigate if we can use retrograde transport to treat genetic diseases. If we could fix the genes by using a viral target, a viral vector, or microRNA to suppress defective genes, if we can get them to where they need to be through this retrograde transport. The myelin sheath, whether it is peripheral or central nervous system, is made of myelin, which is a whitish protein lipid substance. It electrically insulates the axon as well as physically protect it. It increases the speed of nerve transmission. As we talked about on Tuesday, we can get saltatory transmission. The myelinated fibers are segmented sheaths surrounding long, large diameter axons, whereas there are also some that are not myelin covered. Those would be non-myelinated fibers, and they don't have the myelin around them again, so it takes longer for impulses to move down them. So again, in the peripheral nervous system, that myelination is formed by the Schwann cells, which each number of Schwann cells will wrap around in a jelly roll or Swiss roll fashion. One cell is going to form one segment of the myelin sheath. The outer collar of the paranuclear cytoplasm, which we used to call the neurolemma, is the peripheral bulge containing the nucleus and most of the cytoplasm. Plasma membranes have left proteins. There are no channels or carriers, so they're very good insulators, and their interlocking proteins bind the adjacent myelin membranes. So here is what we mean by a jelly roll. The cell grows in this way until it's wrapped around numerous times, insulating and protecting that axon. Those gaps in the adjacent Schwann cells are sites where collaterals can emerge. They used to be referred to as nodes of Ranvier. I guess I should say they traditionally are referred to as nodes of Ranvier. They are now just myelin gaps. The non-myelinated fibers are not wrapped in myelin. They may have Schwann cells, but they don't coil. One cell might be around 15 different fibers. So again, here's our typical motor neuron with its Schwann cells forming the myelin sheath and the myelin sheath gap, which I call the node of Ranvier often. In the central nervous system, those are formed by the oligodendrocytes, not by an entire cell. Each oligodendrocyte can wrap around up to 60 axons at once. We still have the sheath gap present. There's no outer collar or paranuclear cytoplasm. And the thinnest fibers are unmyelinated, but they're covered by long extensions of adjacent neuroglia, so they're not free. So in the central nervous system, we have a setup of what we call white matter and gray matter. White matter is regions of the brain and spinal cord where you have a lot of myelinated fibers. These are usually referred to as fiber tracts. Remember, we talked about tracts earlier. They're white because the myelin has a white color to it, lipid color to it. The cell bodies are going to look grayer. So if we're looking at neuron cell bodies and non-myelinated fibers, that would be our gray matter. So gray matter usually refers to the neuron cell bodies, while the white matter are the tracts carrying information around the central nervous system. And here again are oligodendrocytes. Neurons can be classified into multipolar, bipolar, or unipolar. They're also pseudo-unipolar. Multipolar has 3 or more processes. One of those will be an axon, the rest will be dendrites. In the bipolar, we have 1 axon, 1 dendrite. And the unipolar, we can have pseudo-unipolar, peripheral, or proximal. And they are outlined here. More here. Functional. Pause for a second.